Hello everyone, in this video I'm going to be teaching you depolarization and muscle contraction in the heart. I'll also discuss conduction systems, how signal is spread across the heart, and how the heart will exert force. So firstly, let us go over a resting membrane potential. So what I drew is a cell within the heart. Its normal resting membrane potential would be about negative millivolts. Outside of the cell, we normally have more sodium on the outside and potassium on the inside. This is what causes the inside of the cell to be more negative. When the heart says it wants to depolarize, when it says I want the heart to contract, we have a passive sodium channel in the cell, which is going to cause sodium to go inside of the cell, making the millivolts less negative. It's going to be about negative 55 millivolts. So that is going to cause us to get to the threshold of a action potential, which is where we can have the spike and cause a contraction in the heart. To counteract this, we have the sodium-potassium pump, which is what regulates and maintains this natural gradient of negative inside and positive outside. So let me quickly draw a graph. We have time and millivolts. So just for clarity's sake, this is what the SA node does, and I will go over that in a moment. But what this graph looks like is that we will have negative 70 millivolts, which is normally at our resting. Once we reach negative 55, we will have a big spike up. This will then go down, and essentially this is going to go really down because we're in the negatives, and it will slowly go back up. So here is the sodium channel opening. At the top is when the sodium channel closes. And what's going to counteract that is the potassium channel opening. And this here is called creep, where it slowly goes back to its normal resting state. I wanted to emphasize that the way this occurs is because of intercalated discs. That's a landmark in the histology of cardiac tissue where it will cause this signal to go from cell to cell to cell because it has no resting membrane potential. So since I referenced the SA node, let me draw a heart. It is a potato that sits in the center of our chest, divided into 4 quadrants. We have here the SA node that sits about on the right atrium, and this sends signaling 360 degrees across both the right and left atrium. That is what was exhibited there on that graph, molecularly. So this signaling does not go into the ventricles because we have a wall of dead cells that divides the atrium from the ventricle to prevent signaling to go to both. But what we do have is the AV node, which diverges into the bundle of His, down the bundle branches, and up and around to the Purkinje fibers to cover all of the ventricle. Looking back at the graph, the SA node fires at 100 beats per minute. But as an adult, our resting heart rate is between 60 to 100. So why is it that our SA node is constantly firing at 100 beats per minute, but that does not portray our resting heart rate? That is because the peripheral nervous system, the vagus nerve, innervates both of the SA and AV node. So here we have the PNS. It goes into two branches, one for each. We'll call this branch 1, which innervates the SA node. It will cause, it's called chronotropy, which essentially is the rhythm or the rate of firing. So it is going to decrease because PNS, we've learned, is rest and digest. So it's going to decrease or slow down the rate of the firing of the SA node. We also have branch 2, which innervates the AV node. It's going to be responsible for dromotropy, which causes AV nodal delay. It says it within the name. AV node is going to be delayed from firing because it is going to wait for the SA node to completely finish its firing across both atriums before the signaling occurs for the AV node. This can be seen in a EKG. Hopefully you remember that P wave, QRS complex, and T wave. The P wave is the atrium depolarizing. QRS is essentially the ventricle depolarizing. And the T wave is the ventricle repolarizing or relaxing. So the AV nodal delay can be seen right here at this plateau. It can be seen after the atrium contracts, but before the ventricle depolarizing, just like the SA and the AV node seen here. The way we can electrically read this is because of a EKG, or also called ECG, but because of this graph, consider it as negative and positive. When we put electrodes on a human body, in an EKG there are 12 in total, but I am just going to cover the baseline 3. Right arm, left leg. We have 3 leads, 1, 2, and 3, and our heart would sit about over here. I wanted to redraw the SA and AV node, as you could be able to see the parallel of the plans. So lead 1 follows the heart from right to left. You can see that it follows the plane of the SA node firing. It's in parallel. So that's why you can see that there is a positive increase in the P wave. For the QRS complex, specifically from Q to R, you can see there's a big spike and increase. That is because it is in parallel to the bundle branches going down the septum of the heart. It's going from negative to positive. So it's collecting positives at the end, but vice versa, there's a big spike going down toward negative. That's because of the Purkinje fibers going in the opposite direction from the positive, and you can see the arrow pointing toward the negative. So that is what occurs electrically during the heart. We can now talk about inotropy, which is the process of contraction. This is our lovely goldfish diagram. This is a sagittal view, but I just wanted to emphasize that The first step is the T-tubule having a depolarization signal going down deep into the cardiac tissue. This is a sagittal view, so it is going down into it. Right next to the T-tubule, we have calcium feet, which are sensitive to this depolarization. And right next to the feet is our sarcoplasmic reticulum, call it SR for short, which stores calcium, lots of calcium. We also have these two landmarks, but I will discuss it afterwards, just going in sequence. So T-tubule has a depolarization signal go down deep in the tissue. Calcium feet will sense this signal and trigger the SR to release calcium into the tissue. This calcium is going to go to the sarcomere. So I just drew the Z-band. These blue ones are actin, and this purple one is myosin. Attached to myosin, to anchor myosin to these Z-bands, is titin. And on top of actin, we have tropomyosin. Well, we have troponin, which is consisted of 3 layers on top of the tropomyosin. So let me give you more of an in-depth look. So we have actin band, tropomyosin covering actin, specifically the active binding site. And then we have 3 layers of troponin. We have TNT, which is troponin, tropomyosin. We have TNC, which will bind to calcium. And we have TNI, which is troponin inhibitor. And let's just draw myosin down here with a myosin head. So the myosin head cannot bind to actin because of the tropomyosin blocking it. With calcium, it will bind to TNC, which will cause TNI to move. TNI will move, which will then cause TNT to move. All of this is going to cause tropomyosin to move out of the way for actin to be exposed. So it's just essentially pushed out of the way. So now that it is exposed, myosin head is able to attach itself to the active binding site in actin. Once it attaches, we will have ADP attach itself to the actin head, which will cause a half turn, the cock and pivot per se, and then inorganic phosphate will attach itself also to the myosin head and will cause a full power stroke, which will push the myosin head down and across the actin band. We will then need ATP to bind to the myosin head to release the myosin head so that it can realign itself and reattach so that it can do the cock and pivot and the power stroke to work alongside the band of actin. ATP is made of ADP an inorganic phosphate. So it's actually going to recycle itself. So it will detach the myosin head, use ADP, do half of the stroke, inorganic phosphate, do the full power stroke, and then ATP to release the head, and continuously do that until the Z bands are closer together and the actin is more on top of each other for optimal cross bridges. This can only occur if calcium is present. Without calcium, TNC cannot be bound and it cannot move TNI or TNT to move tropomyosin out of the way to expose actin. You need calcium present for all of it to fully have a contraction in the sarcomere. Once the brain stops sending a depolarizing signal down the T-tubule, it will stop acting calcium feet and stop the sarcoplasmic reticulum to release calcium. But with that stopped, there is still calcium floating inside the cell, which we do not want. So we will have circa be used. It's essentially, circa is a vacuum, and it will suck calcium back into the sarcoplasmic reticulum for storage. We also have phospholamban. Phospholamban is a inhibitor of circa, and it controls the whole storage system. So if you have a low phospholamban, you're going to have low calcium. If you have high phospholamban, you're going to have an inhibited circa. Regarding this inotropy, I wanted to draw another quick graph, which is very similar to the SA node graph. So again, we have MV and time. This graph is going to have a resting membrane potential, but once it's met, sodium is going to open. Sodium channels are going to open. At the tippy top, sodium channels are going to close, but there's going to be a calcium leak, which will cause a decrease, and there's a plateau here of calcium trigger, which is the trigger of depolarization from the T-tubule. That is where all of that has occurred. And lastly, this will decrease and go down as potassium channels open. The main cause of calcium in this is because calcium pores are slower, and therefore it's going to increase the time you will have to complete a contraction inside the heart. So that mainly covers part 1 of the discussion, so we can go into part 2 of heart failure. So heart failure just means that the heart is not able to pump. We have essentially 2 types of heart failure. We have HFrEF, which does not look how it sounds, but HFrEF is systolic failure of the heart. Essentially, that means that there is a reduced ejection fracture, and considering this to Frank Starling law about the greater the stretch, the greater the squeeze, it's going to be reduced overall. So compensation from the heart is that it will add sarcomeres in parallel, but in chronic cases, the ventricle is going to add sarcomeres in series, essentially remodeling itself to dilate and overstretch the ventricle. And there's no cure for that. The second heart failure is HFpEF. This is diastolic heart failure. This is where ventricular filling is impaired. So you are not able to fill the ventricle as much, not able to eject as much out because there's not as much volume. To compensate for this, the heart can increase heart rate so that it can pump more out despite it not having as much volume. But this is counterintuitive because it's going to decrease the fill time of the heart. So it's not going to be able to fill as much as it could possibly eject out. Imaging modalities that we can use is a MRI, which is short for magnetic resonance imaging. MRI is used for soft tissue, but how it works is that a patient is placed on a table and put into a bore, is what it's called, but essentially it's a huge magnet. Normally, the atoms in our body are pointed in all different directions. But when placed inside of a magnet, our atoms are going to point all in the same direction. So what will happen in an MRI is that the patient is put on the table, placed inside the MRI machine in the coil. A radio frequency is going to be shot to the body, specifically where the imaging needs to occur. It will go back to how it originally was, with the atoms being all in different directions, but with it being inside the magnet, it's going to go back in the same direction and correct itself. It's going to realign. This is called precession. And that is where the image is taken. So for the heart, we specifically can use a short axis view MRI for imaging of the heart for HFrEF and HFpEF. So for HFpEF, you will be able to capture this image at N diastolic, where the big stretch is going to occur. This is going to be at Q to R in a EKG. So from Q to R, that will be a big stretch, and you'll be able to see that imaging occur for that failure of diastole. And for HFrEF, you can see this at end systolic, which is right after the QRS complex is completed. So before the T wave occurs, right there. It is not possible to capture this image manually as a human while our heart is pumping. So what we can do is that we can attach a EKG while you're doing a short axis view in the MRI machine, so that it will automatically take it once that point of interest is happening on the ECG. So down here I found a few pictures of a short axis view. This top one, they've already labeled left ventricle, right ventricle, and you can also kind of recognize landmarks of how there's more myocardium on the left than the right, and it's just more of an interesting view. The second picture, you can definitely see in comparison how the left ventricle has ballooned up. So that is definitely the big stretch we were just talking about for HFpEF for end diastolic. And this last one is actually a GIF. This is in motion. You're able to see blood moving. You can see the valves opening and closing, and you can see the contraction and movement of the ventricles and atriums. So just multiple pictures taken at a time to see this movement. It's very interesting. So that's the conclusion of this prompt. I hope you learned something. Well, thank you for watching.